Next we are doing kingdom Monera. Kingdom Monera. Bacteria are the sole members of this kingdom. All the organisms in this kingdom will be called bacteria. There are different types of bacteria. They are the most abundant microorganisms. Most abundant microorganisms. Organisms belonging to the kingdoms Monera, Protista, fungi, they are all called microorganisms. Some organisms in Plantae may be microorganisms and very few forms are present in Animalia. And kingdom Monera of all the 5 kingdoms has the most abundant microorganisms. They are habitat. They are ubiquitous in nature. Ubiquitous meaning they are found everywhere on earth, even extreme conditions. Who will be found in extreme conditions? The archaebacteria. Even extreme conditions like hot springs. They are found in hot springs, snow, desert, deep oceans, everywhere. Next, bacteria are said to be simple in structure but complex in behavior. Very important sentence from NCERT. Simple in structure but complex in behavior. Their structure is simple but their metabolic behavior is very complex. Murti lahan but kirti mahan. Okay? And this one sentence can be twisted in number of ways to create 4 options. For example, a question that has appeared in NEET. Bacteria are dash. Option. Simple in structure, simple in behavior. Complex in structure, complex in behavior. Simple in structure, complex in behavior. Complex in structure, simple in behavior. The correct option will be simple in structure but complex in behavior. Okay? Bacteria have the most extensive metabolic diversity. Most extensive metabolic diversity. They show autotrophic nutrition, they show heterotrophic nutrition and so many different variants within each. So that's metabolic diversity, we will be seeing that. Next we are looking at the 4 categories of bacteria on basis of shape. Bacteria may be available in 4 different shapes. They may be these shape, they may be these shape, or they could be shaped like cashew nuts like this, or spiral shape like this. Actually spiral shape in this manner. Okay, these are spherical shape, these are rod shaped, these are comma shaped, and these are spiral shaped. The spherical shaped ones are called as coccus, singular, plural is cocci. Example, streptococcus. You must have heard of streptococcus. Why is streptococcus called streptococcus? Because it is a coccus, because they are cocci cells. Next, rod shaped bacteria are called as bacillus, singular, bacilli, plural. An example is lactobacillus. Why is lactobacillus called lactobacillus? Because it is a bacillus. Next, comma shaped ones are called as Vibrio. An example is Vibrio cholerae, the bacteria that causes cholera. Finally, spiral shaped are called Spirillum, singular, plural, Spirili. An example is Spirillum volutans, one of the longest bacteria. You can see it with a naked eye. Spiral shaped, Spirillum volutans. So these are the 4 types of bacteria on the basis of shape: cocci, bacilli, Vibrio and Spirili. Now you know kingdom Monera are divided into 2 types, isn't it? Archaebacteria and eubacteria. Archaea means ancient and eu means true. So as such, archaebacteria are also called as ancient bacteria formed on primitive earth, and eubacteria, they are also called as true bacteria and they have been formed on modern earth. Next, cell wall. Cell wall, as you know, prokaryotes, monerans have peptidoglycan cell wall. Peptidoglycan is present in the cell wall of eubacteria. What about archaebacteria? Do they also have peptidoglycan? So they have pseudo-peptidoglycan, which is pseudo or false, okay, which is chemically similar to peptidoglycan with some differences. It is chemically similar to peptidoglycan with some differences. Peptidoglycan has two sugars in it, NAG and NAM, wherein pseudopeptidoglycan has the two sugars NAG and NAT. Find out what is NAG, NAT and NAM. Do research. Find out the names of these short forms. Other than the cell wall, are there any other differences? So let us say this is the bacterial cell. The outermost layer is the cell wall, right? What is there inside the cell wall? There is plasma membrane. If I take a part of this plasma membrane and put it over here, I will see a structure like this. Such kind of molecules are seen, which are lipid molecules. There is a sea of lipid molecules, and few will be protein molecules floating in the sea of lipid molecules. The lipid molecules, as you see, are in two layers. This is called a lipid bilayer. The membrane is said to be having a lipid bilayer structure. This kind of a structure of plasma membrane is also seen in plants and animals, including us. What about archaebacteria? Do they also show the same structure of plasma membrane? Let us have a look. So, their structure will be in this manner. What do you observe? Is there a lipid bilayer? No, there is a monolayer. There is a monolayer of branched chain lipids is what you see in case of archaebacteria. So this is about their plasma membrane. Look at the differences between archaebacteria and eubacteria in terms of cell wall and plasma membrane. Carl Woese, you can make a point, Carl Woese considered differences in cell wall, cell membrane and 16S rRNA sequences to divide Monera into two kingdoms, out of which cell wall, cell membrane, I told you the differences. There are differences in this thing called as 16S rRNA sequence as well. But right now I'm not telling you what is that 16S rRNA. We will come to know about it in future. But this is what he considered for dividing Monera into archaebacteria and eubacteria. Under archaebacteria we will be learning methanogens, halophiles, thermoacidophiles. Under eubacteria we will be learning bacteria, cyanobacteria, mycoplasma. So let us first start with eubacteria, and under eubacteria let's start with bacteria, with the bacterial cell structure. So we are doing bacteria now, and right now we are looking at the bacterial cell structure. Let us say this is a bacteria. If you remember, the cell of a bacteria is simple in structure but complex in behavior. So we are looking at this simple structure now. This is the outermost covering of the bacterial cell. This will be a cell wall. As you know, they have a cell wall. Is it made up of cellulose? No. What is it made up of? Made up of this substance called as peptidoglycan, which is made up of peptide also and glycan also. Peptide is protein and sugar. Protein and sugar. This compound is made up of protein and sugar, and it's called peptidoglycan. This peptidoglycan arrangement can be of two different types, okay, or what I have shown you is a small portion of the cell wall. So as you can see, the cell wall may have only peptidoglycan, a thick layer, okay, or else it may have, okay, or they may have a peptidoglycan layer which is thin and another layer outside which is made up of lipopolysaccharide, also called as LPS. And based on this cell wall arrangement, we can say that bacteria are of two different types. या तो bacteria have this type of cell wall या फिर this type of cell wall. The ones that have only peptidoglycan are known as gram-positive bacteria, and the ones that have peptidoglycan along with LPS layer are called as gram-negative bacteria. Why are they given this name is that the gram-positive bacteria are able to retain one stain called as the gram stain, which is violet in color. If you pour that stain on these people, they will be able to retain that stain violet in color, and these cannot retain this stain because of the presence of the LPS layer. Okay, they will look pinkish in color. Okay, so you have to say that they retain gram stain and they do not retain gram stain. Why this is necessary to know is that medicines to a patient will be administered on the basis of whether the disease causing bacteria is a gram positive bacteria or a gram negative bacteria. That you must know first before you administer medicine, because it needs to penetrate these particular types of cell walls, gram positive or gram negative. Okay, irrespective, the cell wall is made up of peptidoglycan in bacteria, unlike any other kingdom. Okay, so that is about cell wall. Some bacteria may have another layer outside the cell wall, and this will be present in some bacteria only. This additional layer is called as Glycocalyx, present in some. If it is present, it may be present in two forms, capsule or slime layer, wherein capsule will be a tough layer and this slime layer will not be a tough layer, it will be a smooth slippery layer, and it is meant for attachment. This one is meant mainly for protection. This is the tough layer, this will be slimy in nature. This one is meant for protection, this one for attachment. So these are the outer two layers of the bacterial cell. Inside the cell wall, there will be a plasma membrane which will be enfolded in certain places in this manner. One major enfolding will be present. I'll tell you about that later. So this third layer inside is the plasma membrane, and these enfoldings are called as mesosomes. Plasma membrane, the cell membrane that is present outside every cell including animal cells. Okay, and the enfoldings are called mesosomes. Enfoldings of plasma membrane. One of these mesosomes holds on to the genetic material inside and helps it in duplication process. We will see about that later. Okay, inside the cell then this is all cytoplasm. So this is all cytoplasm here, and inside the cytoplasm in the center of the cell there will be this. This is a single circular chromosome called as the nucleoid. Now what is this? In animal cells you have, this is a typical animal cell with plasma membrane. In the center you will see the nucleus, and inside the nucleus we have 23 pairs of chromosomes, that is 46 chromosomes in total. Okay, over here in contrast you see this is a very primitive cell, by the way, so that's why it does not have so many advanced structures like our cells. There will be just one chromosome, like how we have 46 chromosomes, they have just a single chromosomes with all the genes required to maintain this cell. Alright, a single chromosome, okay, of double-stranded DNA, and it is circular in nature. Why I am saying circular? Because in our chromosomes are linear in nature. Okay, the ends are not connected to each other in a loop. Over here it is a circular double-stranded DNA, no histone proteins. Why I am saying no histone proteins? Because our DNA is folded using histone proteins. Histone proteins are the proteins around which our DNA is wrapped. In eukaryotes, they do not have histone proteins. And this single circular chromosome is called a nucleoid. And because they have just a single chromosome and no other chromosome, a double copy, because our every chromosome has another copy just like that one, just like itself, right? That's why we say that we have 23 pairs of chromosomes, because for each chromosome there is another one exactly like that one inside the nucleus, and as such we have 23 such pairs, okay, inside the eukaryotic cell. But in bacterial cells they have just a single chromosome, and that's why they are called haploid. And that is what makes them haploid organisms, haploid or N. And we are diploid or 2N. Okay. Next, in the cytoplasm you will see a number of structures, granular structures, which are called ribosomes. What are ribosomes? They are the protein synthesizing machinery of the cell, right? Ribosomes are involved in the formation of proteins of the cell, and they are scattered throughout the cytoplasm. They are 70S type and they are said to be scattered. Why I am saying scattered? Because in eukaryotic cells the ribosomes are located only on the endoplasmic reticulum. Okay, you know endoplasmic reticulum, right? They are located on the endoplasmic reticulum. Very few will be present in the cytoplasm, but over here all are present in the cytoplasm. No membrane bound organelles will be present inside this cell, like how you see in the eukaryotic cell. In eukaryotic cell you will have mitochondria, you will have the endoplasmic reticulum, you have chloroplast, vacuoles, etc. These are very primitive cells. They do not show any type of membrane bound organelle. Okay, no ER, no mitochondria, no nothing. Just there is cytoplasm and the nucleoid. Clear? So we say there are no membrane bound organelles like ER, mitochondria, etc. Ribosomes are 70S type ribosomes, telling because the ribosomes in eukaryotes are 80S type ribosomes. Okay, this is just for reference I have given eukaryotic cell. If you want we can make a note. Eukaryotic cell, 80S type ribosomes, membrane bound organelles present. What else? They are diploid, 2N. They have linear double-stranded DNA. Mesosomes are absent. So this is a very primitive type of cell. It has come very earlier into the evolution process. That's why it does not have anything much. It's very simple. And later on, as the bacterial cells started advancing, they started having a nuclear membrane around this that made them eukaryotic in nature. The first protestant cells were formed, and other membrane bound organelles started forming that formed a different organelles, what you see in the protista. And from protista then they further evolved into plants. Some protists evolved into animals, some protists evolved into fungi. Some bacteria may show the presence of small circular DNA floating into the cytoplasm other than the main nucleoid. These are called as plasmids. These are said to be extra chromosomal DNA particles which can provide them extra characters like resistance to certain antibiotics, etc. If they have them, they will be advantageous to them, mostly. Alright? Next. This is a flagella. What is flagella? It is an organ for locomotion. The ones that have them will be motile. The ones that lack a flagella will be non-motile in nature. E. coli, the very famous bacteria, has many flagellae. So it is very motile. It is made up of a protein called flagellin. Made of flagellin protein. Next, such hair-like structures may be seen in some bacteria. These are known as Fimbriae. Fimbriae are meant for attachment. Some bacteria may have a structure like this, a short, sturdy structure, which is called a pilus. Pilus is like an organ that makes this a male bacteria. So you can infer what its role will be like. If this makes it a boy, and if a bacteria does not have it, makes it a girl bacteria. So this is a pilus, and it may have a role in sexual recombination, transfer of genetic material from one bacteria to the other bacteria, reproduction. Okay, has a role in sexual recombination, about which you will learn later. Mesosomes over here are involved in two roles. Okay, first role is they are involved in binary fission, and the second function that they have is they have the enzymes inside them for cellular respiration. Since they do not have separate organelles like eukaryotic cells, like mitochondria, okay, where are they going to carry out their cellular respiration? So they are going to carry that out inside the mesosomes, on the membrane of the mesosomes. So write down involved in cellular respiration, and they are similar to mitochondria. So with that we have completed the bacterial cell structure.
महादेव के नाम पे सब्सक्राइब कर दें